This is the pathway known as glycolysis. It's the pathway by which glucose gets broken down into, in this endpoint here, pyruvate. And from here, it's a two-step jump to ethanol and carbon dioxide. So this is essentially what the Buchner brothers were seeing in their mishmash of cell extract here. They didn't know it at the time, but these enzymes of glycolysis were in this cell-free extract, converting whatever glucose was present into fermentation products. And it didn't take too long, decades after them, for people to isolate these players, things like aldolase, these other enzymes, and study their properties and figure out exactly what they do. We will spend time on glycolysis. Another kind of question that some biochemists would look at is just how things are built and what holds them together. So here's an example of a so-called alpha helix in a protein. People, mostly X-ray crystallographers, purify biological molecules, take molecular pictures of them, basically through X-ray crystallography, and figure out what's where, how it's connected. Is it a covalent bond, or is it one of these dotted things that's a hydrogen bond? The structure is very important, and the first step in understanding something like an enzyme maybe is to understand how it's built. And that plays into this concept. Many, many people in all aspects of biochemistry are interested in this. How does the structure of a molecule, particularly a macromolecule, relate to the function of that macromolecule? So that protein graph that we saw on the last page has a particular structure. How does that play into the function of that protein? And this is nothing more than what biologists have been doing for centuries. Like early anatomists would cut open an organism, that cut open an animal and see lung tissue, and they get under, once microscopes were invented, that start looking at the details of the lung, and they would see cavities and air spaces and things. And it was clear that this was a structure designed for gas exchange, or as they'd look at kidneys and see all kinds of intricate ductwork and plumbing that related to the function of a kidney and excretion. So you could look at the structure and know something about the function. Biochemists do the same things with macromolecules. So, for example, this is a protein DNA mix. You'll recognize the double helix in here. On each side there is this protein called a TUS protein. People had found this protein, weren't quite sure what it did. They crystallized it, co-crystallized it, in fact, with DNA strands, and they found that this particular strand of DNA. The geneticists were kind of thinking that it had something to do with regulating the cycle of cell replication. It turns out that this TUS protein binds to specific sequences of DNA, specific sequences of monomer nucleotides in this DNA, the gene sequence, if you will. And this protein has a structure that is complementary enough that it wraps itself around the double helix, and it can find these grooves in it and pack in very tightly through weak interactions. And it's not just binding to any old piece of DNA. This protein has specific interactions with specific bases in the gene sequence. So it only binds to this stretch of DNA. Out of the miles and miles of DNA, it comes along and it recognizes that one. It reads the DNA sequence, basically. So you can talk about the function of the molecule. You can talk about the function of individual amino acids on the protein that must be interacting with specific bases. And so it's an understanding function by understanding the structure.